We'll begin to look at the biochemistry of the body. And so essentially, when we talk about biochemistry, we're talking about the organic molecules that are going to function within the body in different formats and different processes within the body itself. And so we have our macromolecules that we're going to start to look at. And within them, we have our basic types. We have our carbohydrates, we have our lipids, we have proteins, and then we have nucleic acids. And so each one of those is derived from a smaller structure, and then we build up the larger structures to within them. And so we have both polymers and we have monomers. And so these polymers and monomers are in different sizes and different numbers of the building blocks for each one. So things like carbohydrates, we think of as sugars. And so these are monosaccharides. These are monomers of the sugars themselves. And so these are the reduced forms of all of the sugars that we can have. Lipids derive from things like fatty acids and monoglycerides. And so we have kind of two different ways that we can start to put them together within them. And those are going to build up into larger lipids, the polymer. Amino acids will bring together to make us proteins. And so we'll take a look at those as well. And then lastly, nucleic acids to build up things like nucleotides and high energy sources as well within the body. So carbohydrates themselves, we have one of our essentially food sources that we have there. And within that, primarily we utilize carbohydrates for things like energy storage. And so we're going to store those sugars away somewhere in the body itself. But at the same time, we use it for our everyday kind of energy sources. So we're going to utilize all of those sugars that are stored in all of the foods that we have there and be able to break them down and break the bonds within them to release the energy that is stored within those bonds. And so that breaking of those bonds is what's going to allow us to create the energy that we need for the bodies themselves. In addition to being an energy source in the body, they do become part of the structural component of the cells themselves. And so they become part of the plasma membrane. They become part of the internal structures of the cells that we have. So when we look at carbohydrates, essentially some of our most simple sugars are things like our glucose. And glucose is going to be the primary cellular fuel that we have for the body itself. And so this is what we're primarily going to use for energy within the body, is this glucose here. And so carbohydrates, oftentimes abbreviated as CHO, because they always contain a carbon, a number of hydrogens, and a number of oxygens within them. And so the ratio is going to change depending upon the particular type of sugar that we have and the different type of carbohydrate that we have in the end as well. And so we can base that number and get us the final amount of each one that we have there. Carbohydrates themselves do become fairly lengthy in terms of their structures that are there. And in our own bodies, we utilize glycogen as an energy storage form. But in things like plants, they're going to store energy as either starches or cellulose. And so they're going to store that there within them. Our own bodies, when it comes to eating things like plants, we can't break down cellulose, and so we're unable to break down those sugars that are there. And so those are going to go through the digestive process without having been broken down because we just don't have the enzyme in order to break them down. Starches, on the other hand, we can definitely break down, and we can utilize their energy that they have there for us, but we can't liberate any energy out of the cellulose. When we have excess amounts of glucose within the body, we're going to take those excess amounts of glucose and combine them together to give us glycogen. And so we're going to make long chains of glucose molecules, and we'll take the individual glucose molecule and we'll bond them together with another glucose molecule and another glucose molecule until we have this big long structure of glycogen. Multiple branches on them, lots of side chains that are coming through them. That's creating this big long storage that we can store away in the body. And so the places like the liver and the skeletal muscle store a large amount of glycogen within them for use when they need them. So when we have excess amounts of that glucose, we can store it away in a sense for kind of like a rainy day. Lipids are next, and with lipids, we have a higher energy stores. And so we do once again have energy storage capabilities within lipids, and we increase the number of, or the amount of energy that can be liberated from lipids quite dramatically compared to that of carbohydrates. And so they're a high energy source for the body itself. But primarily, we're going to use those carbohydrates as our main source of energy the majority of the time. Rather than using lipids for energy on kind of a daily moment-to-moment basis, we're going to find them in places like the cells. And so the lipids are going to make a large component of the cell membrane. And so we're going to have a large amount of lipids combined with other structures in the cell membrane to give us the overall structure that we have there to protect the cell itself. In addition, we can use components of lipids, especially things like steroids, in the production of hormones. And so we can produce our steroid hormones themselves from these lipids and generate the ability to communicate within the body itself. Once again, when we look at lipids, we again have monomers and then we have polymers of lipids themselves. And as they combine together in a couple of different ways. And so you've probably all heard of things like saturated fats and unsaturated fats, and you look at them and kind of see what they have going on. And we see that in terms of the types of fats, we don't want the saturated fats, we want the unsaturated fats. And what it comes down to is the carbon bonds. And so in saturated fats, a carbon molecule, every carbon molecule that we have along this particular lipid here has a single bond connected to it everywhere. And so it's either connected to a single bond to a carbon or to a hydrogen. And so it has those single bonds all the way through it. With an unsaturated lipid, we're going to generate a double bond there. And so that double bond is going to change the way that the molecule itself lines up. And rather than going from a nice linear appearance as far as that molecule goes, we now have a bend. And so it has bent essentially that structure that's there. It's relatively easy to determine whether a lipid is a saturated or an unsaturated type of a fat just simply by looking at it. And so when it's sitting there at room temperature, what does it look like? Is it solid form like butter, or is it liquid form like vegetable oil? And so it has that component in there changed, and that changes the properties of the molecules themselves. And so depending upon whether we have that saturated carbon bonds that are there or not is going to dictate whether we have something that can remain solid at room temperature or something that's going to turn into a liquid once it's there. So when we look at lipids themselves, they again come in multiple varieties as far as the types that we have there. And so inside the body, we have things like triglycerides. And so you go and you get your blood tested. Part of what they're going to tell you there in terms of the fat panel that you have, the lipid panel that you get, is going to be that you have triglycerides. So you'll see things like triglycerides listed. You'll see things like cholesterol listed as far as the different types that we find there. And for each of them, we do need them within the body. And so triglycerides we're going to see as fats and oils within the body itself. Steroids we're going to use as things like cholesterol. And so that cholesterol makes up, in terms of the cell membranes, as much as about 20% of a cell membrane can be made up of cholesterol. And so we do need a large amount of them in those cell membranes themselves. Different sex hormones, especially the male sex hormones, are going to utilize that cholesterol and those steroids for that process as well. Every single cell that we have in the body also contains phospholipids. And so we've got phospholipids in the cell membrane. And so we have this phospholipid here. It has two major components to it. It has a head, and then it has a tail. And the head and the tail are different in terms of how they interact with other molecules. And so we have hydrophilic heads and hydrophobic tails. Hydrophilic meaning that it likes water, hydrophobic meaning that it doesn't like water. And so we can utilize that with, say, our cell membrane, where we have water on the outside of the cell, and then we also have water on the inside of the cell. So those heads and tails are going to point in opposite directions in one another. So inside our cells, in that plasma membrane, we're going to have a set of heads and tails that are in two layers. And so the heads are going to point towards the outside of the cell or the inside of the cell, and then the tails are going to point to one another as they don't like water there. We can also use it rather than in a cell membrane. We can also use it in structures called mycelials within the body as well. And we can use them to help us to transport substances. And so here we have those phospholipids, where we have our hydrophilic heads pointing towards the outside. We have our hydrophobic tails on the inside once again. And so we can put things like lipids inside there and transport them throughout the bloodstream and not have them affected by the water that are there. So they're going to kind of come together when we do put them within a water structure. Next up, we have proteins. And so proteins themselves get somewhat complicated as far as their overall structure goes. But proteins are a very dynamic set of structures that we have in the body. They do a lot of different things for us. So they can be actually parts of things, and so they can be structural. And so they can be parts of our hair, our skin, our muscles. Proteins are kind of everywhere within the structural components themselves. They can help to regulate sizes of things in the body. Other proteins are things like hormones, and so they can be regulatory in themselves. So we can use them as a messenger in a sense. Certain proteins are utilized in the body for help with transport. And so inside our red blood cells, we have hemoglobin, and this is how we're going to transport oxygen within the body itself. We can transport other molecules as well by binding them to a protein. So some of those hormones that we have need to be bound to a protein in order to transport. And so we have the two of them kind of combining together. Other places, our immune system, we have antibodies. And so this is going to prevent us from getting infected once again from a particular infection that we've come across in the past. So they're going to become part of our immune memory as to the things that we've reacted to in the past that's there. And in places like the digestive tract as well as elsewhere in the body, they serve as enzymes. And so these are enzymes that are going to be catalyzing chemical reactions. So they're going to speed up those reactions to allow them to go faster than they normally would. So proteins are a pretty important thing that we have within the body itself. And they have basically 4 different levels of structure to them. And so if we were just to kind of, in a sense, draw out a particular protein or write down all of the amino acids that are there, we would have this big long chain or this long string of amino acids. And so here we have just kind of a generic listed as just A1, A2, A3, A4, A5, and so on. We have this big long list of amino acids that make up this protein that is there. And those are going to be individual amino acids. We put them in the correct order and we get a particular protein. If we change the order in any way whatsoever, we're going to get a different protein. It's kind of like a big recipe. And so we have this recipe where we're going to put individual amino acids together. We put them in a particular order and we get a particular protein. Versus if we change that order just a little bit, we get a completely different type of a protein altogether. Just based upon those amino acids. And so, as I said, it's kind of like a recipe. If we take out this ingredient and put in that ingredient as we're baking something like that, we may get a loaf of bread that is all flat and hard and dense, or we may get a loaf of bread that is all big and fluffy and things like that. Or maybe we change it up a little bit more and we add another ingredient into it, or maybe 2 more ingredients, something like that. And now instead of bread, we've got cake. And so those kinds of changes to the recipe make a completely large change into the overall end product that we end up with. So in terms of those levels of kind of packaging our protein up, we start out with our primary structure. And that primary structure, again, is just that linear component of amino acids. And so we just look at essentially all of the amino acids that are there. Our secondary structure lines up our proteins in either beta-plated sheets or alpha helixes. And so depending upon how the bonds come together with the hydrogen bonds is going to dictate how they put themselves together. So we either get these helices, and so nice spiral on them, kind of like a spiral staircase, or we get these bent kind of pleated sheets. Depending upon the particular protein and the different structure, when we then start to fold them in on one another again, we get this tertiary structure. And that can be made up of beta pleated sheets as well as helices. And so here we have our helix again. Here's some more beta plated sheets. We can have those different structures that are there. And then the last component that we'll talk about is the quaternary structure. And so this is kind of our final folded protein structure that we're going to have as multiple polypeptides kind of start to come together to fold within one another. So again, our primary structure is just a linear sequence of amino acids. And so we have one amino acid after another amino acid after another as we see them there on the chain itself. And so it's kind of like a pearl necklace or something, or a diamond necklace or something like that. We have all kinds of individual amino acids or beads that are on there, and those form this particular structure that's there. And so we're going to have the different components that are along there, and they're in their particular order, and we can follow them along, and we can basically, in a sense, kind of write down our recipe. We kind of show them as they go on the structure itself. Our secondary structure, again, brings us to alpha helices and beta sheets that are there. And so our beta pleated sheets that we can see there. And depending upon how those hydrogen bonds combine with one another dictates the overall shape that we end up seeing there. And so we have two major structural patterns that we see with our secondary structure for them. And it's all in the hydrogen bonds that we see as far as the overall structure that we're going to end up having within our protein itself. The tertiary structure now becomes a little bit more complex. With our tertiary structure, we end up having two different types of tertiary structures. We either have globular or we have fibrous. And these are going to bring about essentially two major different kinds of proteins that we see with them. Globular types of proteins are oftentimes going to be things where, like down here in this one here we have hemoglobin. And so here we have the hemoglobin itself, and that globular type of a protein structure is going to allow us to transport oxygen within red blood cells. And so that's going to be bound within the structure there itself. Versus fibrous proteins are going to make up things like collagen fibers within the body. We're going to use them for structural kinds of components within the body itself. And so they're going to be strong. They're going to be able to withstand some stress and things like that as far as the overall protein itself goes. Lastly, we have our quaternary structure. And so with these, as we start to put polypeptides together, those polypeptides, as they start to form with one another, now here once again we have our hemoglobin, and so we have 1, 2, 3, 4 different peptides that come together, and as they come together, that interaction between the polypeptides dictates our final shape of the protein itself. Here in this particular one, essentially 3 strands of proteins are twisting and turning around one another to form essentially a protein band as it comes together within there. And so we have almost what's something that looks like a rope within the protein itself. And that's, again, going to be in things like our collagen fibers that we have within the body itself. So we go from our primary structure over to our secondary structure. We get our alpha helices and our beta pleated sheets. From those, we can then make up things like our tertiary structure, where we now have either globular structures within there where we're binding them together, and then bring those together over into multiple polypeptides. And now we have things like our collagen again and hemoglobin as they come together. And so now we can interact them with one another. And even in things like the hemoglobin here, as oxygen comes in and binds to the iron in the center there, that's then again going to further change the shape of that hemoglobin as well. And so as other molecules, as other atoms, things like that come into play, they can start to change the overall shape once again. In addition to other things binding to those proteins to cause them to change shape, we can do things or change the environment of those proteins to change the shape as well. And so different things can occur to change proteins overall in terms of their appearance. And so here we have, as an example, temperature changing proteins. And so the white of the egg appears to be two completely different things. In the freshly cracked egg, the white of the egg is completely liquid. It looks more yellow in a sense for the most part than it does looking rather white for it. And it doesn't hold a shape at all. It's essentially liquidy as far as the proteins are concerned. But as we add heat to it, though, that heat comes in and it changes the protein altogether. And when that happens, this is what we call denaturing. And so we've denatured the proteins that are there. And as such, we now go from a liquidy kind of yellow structure into something that is solid and white. And so the structures that are there have undergone change. These are permanent structures. We can't take it back. We can't somehow, in a sense, cool this egg off and bring us back over to a liquid state. We just can't do that. And so this is permanently changing what has occurred to that protein altogether. In addition to things like heat, we can change things like the pH of the environment itself. We can change the overall environment. We can add things like alcohols to them, and that can change their overall function within the body as well. And so we'll look at, when we do the lab, we will also look at how environmental changes can change proteins' functionality when it comes to things like enzymes. Nucleic acids, and getting to our nucleotides. And so nucleic acids bring about our ability to store information within the cells themselves. And so cellular function-wise, nucleic acids and nucleotides help in genetic information. So we're going to store information within them, and it comes in two varieties, either deoxyribonucleic acid or ribonucleic acid. And so two different kinds and two different uses for them. DNA is going to be the genetic material that has all of our chromosomal information. This has all of the information for making proteins. And from DNA, we're going to elicit the transcription of RNA. And so we're going to use that to make RNA. And once we have RNA, we can then use that RNA to make proteins. And so it's going to serve as the recipe, so to speak, in order for us to get the proteins that we need. In addition, we can have some of our nucleic acids utilized for, or nucleotides used for energy transport. And so we talk a little bit about ATP as well. So when it comes to nucleic acids, we have adenine, cytosine, thymine, guanine, and uracil. The first four are found on DNA. And so adenine, cytosine, thymine, and guanine are all found on DNA themselves. For RNA, we're going to get rid of the thymine and utilize uracil. So when we look at the two differences there, we have adenine in both, we have cytosine in both, and we have guanine in both of them. But when we have DNA, we're going to have our thymine, and when we have RNA, we're going to have our uracil. And so there's going to be two different components that are there. And so it's one way to be able to kind of recognize, are we looking at a DNA strand or are we looking at an RNA strand? And so from that, anytime that we see uracil, we're going to know that since we have uracil there, that we must be looking at RNA. And so we can see the differences there. That being said, that we have adenine, cytosine, thymine, guanine, and uracil, when we combine them together, especially when we're combining them together in DNA like we have our helix here, they can only combine together in particular ways. And so with that, thymine and adenine can only make a bond with one another. Cytosine and guanine can only bond to one another. And so as we go through here, we can see that those particular ones only bond with their complementary nucleic acid. And so they have to be the right one in a sense in order for them to bond together. So for all of the genetic variation that we have, it comes down to 4 nucleotides. And within those nucleotides, we then only have the ability to combine adenine and thymine and cytosine and guanine, and that's it. Or guanine I just lost my thought. Adenine and uracil in RNA rather than adenine and thymine in DNA. So we can see that we have a couple of different types of nitrogenous bases as well. And so we have purines, and then we have pyrimidines. And so two different components as far as the types that are there. Adenine and guanine always have to bind with their appropriate complement of the pyrimidines. And so a purine is going to bond to a pyrimidine. And so adenine comes down here and bonds to thymine, and it also bonds to uracil. Guanine comes in and bonds to our cytosine. And so one of the things that we can look at is that we have, in terms of our purines, is we have 2 molecules here, which both have a double ring on them. And then our pyrimidines only have a single ring. So when they start to come together, we can kind of think about them as being kind of 2 different sizes. And so we've got to always bond something that's longer with something that's shorter. And so that's going to go all the way through for our DNA structure itself, or the RNA structure that is there. And so once again, uracil here, so we're looking at RNA on the left-hand side. So we have our uracil. It would be looking for an adenine to be able to come together. And if we just look at this here, we see that our uracil and our adenine come together as far as the overall size goes. So we end up with 3 bonds there. And then for that of our guanine and our cytosine, same thing. We have 3 rings as we come together as well. And so adenine and thymine ends up with a double bond. Cytosine and guanine ends up with a triple bond. And so between the two. And so we can kind of utilize that in other processes to be able to determine the number of adenine to thymine and the number of guanine to cytosine bonds that we have. And so that's going to give us a good idea for how big or how long that DNA is and the types of nucleotides that we actually find within that particular organism or that DNA that we're looking at. So we said that we had the ability to transfer energy with our nucleotide as well, and that's where adenine comes into play. And so we're going to take that adenine that we have for our nucleotides, and we're going to be able to combine that with ribose, and that's going to get us adenosine. So adenine plus ribose gets us adenosine. We're going to bond that to a phosphate group, either 1, 2, 3, and we're going to have adenosine triphosphate, or ATP. This is going to be our body's universal energy source. This is what we're going to have for our body to utilize for energy in all of our cells. So we're going to take any kind of carbohydrates or proteins or lipids, and we're going to break them down and liberate them as ATP. So we're going to have this energy available to our cells as ATP. But it all, in a sense, starts with that adenine, similar to what we have on our DNA and RNA. So adenosine triphosphate is a triple phosphate group, and so it does have 3 phosphate groups into it. And there's energy stored in each one of those phosphate bonds. And so if we were to take one of those bonds off, break the ATP, we're going to liberate energy, and the ATP is going to be broken down into ADP in inorganic phosphate. And so we're going to have the 2 of them come off as byproducts of the process. And so we get our ADP here, our inorganic phosphate, and energy when we go in this direction. And so when we break that ATP down, we get inorganic phosphate, ADP, and energy within the cell itself. If we go back in the opposite direction, so if we then take ADP that we have, in a sense, inside the cell, we combine that with some energy that we have from, say, glucose in its bonds that it has there, and that's going to allow us to take that phosphate group and bond it back to the ATP. And so that's going to, or back to the ADP. And so that then generates our ADP plus P gives us ATP, so our adenosine triphosphate, and then water as a byproduct as well. So this allows us to go from things like carbohydrates or our glucose and take the bonds that are there and the energy that's stored there, and rather than taking that energy all in one big huge process, lots of energy released, we're going to take all of that glucose and break it down very slowly, and in that process we're going to make smaller amounts of energy come out, and those are all going to be stored on ATP, and so on. And so we continue around and we add more and more and more ATP there. And so we've got a large amount of ATP available for doing things like muscle contraction. So when muscle contracts, all of the individual proteins that are inside there need just an enormous amount of energy in order for them to contract. And then essentially it's going to power everything else in the body as well. So here, just kind of a breakdown of what we have. So we have our adenine, we have our ribose, that gets us, combine those 2 together, and we have adenosine. We're then going to take adenosine and a single phosphate group, and we would have AMP, or adenosine monophosphate. Add that second one, and we end up with our ADP. Add that third one, and we get ATP. As we go from our adenosine to AMP to ADP to ATP, the amount of energy available goes up. And then once we break one of those bonds, the amount of energy available goes down. And so the number of bonds versus the energy that is available changes as we look at the graph there. So the more bonds that we get, the higher the energy it goes, and then as soon as we break one of those bonds, our energy comes back down with those. So this should be a good introduction into kind of the macromolecules of the body, and we will see you next time as we continue on into the cell.